Genetic drift. Genetic drift is a mechanism of evolution that refers to random fluctuations in allele frequencies from one generation to the next, particularly in small populations. Unnatural selection, which is a non-random process where alleles increase in frequency due to their advantageous effects. Genetic drift is entirely random and occurs due to chance events. One of the key mechanisms in the evolution of populations and plays a significant role in reducing genetic diversity, especially in small populations. 1. Causes of genetic drift. Genetic drift occurs due to random sampling of alleles in the population's gene pool. This randomness can be influenced by several factors. Small population size. In small populations, the likelihood of certain alleles being passed on by chance is higher. Even if the allele is neutral or does not affect fitness, its frequency can change over time. Bottleneck effect. This occurs when a large portion of the population is suddenly reduced due to environmental factors such as natural disasters, disease, or habitat destruction. The surviving population has a smaller gene pool, and as a result, genetic variation is reduced. The alleles carried by the survivors may not be representative of the original population's genetic makeup. Founder effect. This happens when a small group of individuals leaves a larger population and establishes a new population in a new location. The population is likely to have different allele frequencies than the original population simply due to the small sample of individuals that founded it. This can lead to significant genetic differences between the two populations. 2. Effects of genetic drift. Genetic drift can have profound effects on the genetic structure of populations. Loss of genetic variation. Over time, genetic drift can reduce genetic diversity within a population. Rare alleles can be lost by chance, particularly in small populations. Fixation of alleles. In small populations, genetic drift can lead to the fixation of alleles. This means that an allele's frequency becomes 100%, and it is the only allele at that genetic locus in the population. Once an allele is fixed, it cannot be unfixed unless new mutations occur. Increase in inbreeding. As genetic drift leads to a reduction in genetic diversity, inbreeding can increase. Inbreeding reduces heterozygosity and can increase the expression of deleterious recessive alleles. Random fluctuations. Allele frequencies in the population will fluctuate randomly from generation to generation, and some alleles may become more or less common due to chance rather than natural selection. 3. Genetic drift in small birds. Large populations. Genetic drift has a much more significant effect in small populations due to the smaller number of individuals contributing to the next generation. In such populations, random fluctuations in allele frequencies can result in rapid changes in the genetic composition. The smaller the population, the more pronounced the effects of drift. Large populations. In larger populations, the effects of genetic drift are less pronounced because there is a greater number of individuals contributing alleles. As a result, random fluctuations tend to be averaged out, and the population remains genetically stable over time. 4. Mathematical representation of genetic drift. Genetic quantified using effective population size, NA, which represents the size of an idealized population that would experience the same amount of genetic drift as the actual population. The NA is generally smaller than population size due to factors like unequal sex ratios, fluctuating population sizes, and variations in the reproductive success of individuals. The change in allele frequency due to drift can be modeled by the equation delta p equals 1 to n dot operator p dot operator 1 minus p delta p equals 2 n1 dot operator p dot operator 1 minus p where delta p is a change in the frequency of the allele n is the effective population size p is the frequency of the allele in the population. This equation shows that the rate of change in allele frequency due to drift is inversely proportional to the population size. As n increases, the impact of drift decreases. The role of genetic drift in evolution. Genetic drift is particularly important in the evolution of small populations. While natural selection can drive adaptive evolution, genetic drift can lead to changes in the gene pool that do not necessarily confer any adaptive advantage. Drift can also lead to the fixation of neutral or even deleterious alleles, especially in isolated populations. In contrast to natural selection, which is deterministic and directed by the environment, genetic drift is random and can result in evolutionary changes that are independent of the environment. Over time, this redistributes speciation and differentiation between populations, particularly in isolated populations where drift acts more strongly. 6. Bottleneck and founder effects. Bottleneck effect. The bottleneck effect occurs when a population is dramatically reduced in size due to an environmental disaster or other event. The surviving population is often genetically unrepresentative of the original population, and genetic drift can lead to a loss of genetic diversity. This can result in the fixation of certain alleles, even if they were rare or neutral in the original population. Example, the cheetah population is an example of a species that went through bottleneck. The population was drastically reduced at one point, leading to a genetic bottleneck that resulted in a loss of genetic variation. Founder effect. Founder effect occurs when a small group of individuals from a larger population colonizes a new habitat. Genetic variation in the new population is limited to the alleles carried by the founders. Over time, genetic drift can cause significant divergence between the original and new populations. Example, the genetic variation in island populations, such as those on the Galapagos Islands, is often influenced by the founder effect. Many of the species on the islands evolved from a small number of colonizing ancestors and have distinctive genetic traits due to drift. 7. Press questions and answers. Question 1, Gate 2016. In a small population, the frequency of an allele is 0.8. If the population undergoes a bottleneck, what is the likely effect on the allele frequency? A. The allele frequency will increase. B. The allele frequency will decrease. C. The allele frequency will remain the same. D. The allele frequency will be random. Answer. The correct answer is D. The allele frequency will be random. Due to genetic drift, the allele frequency may fluctuate unpredictably in small populations, especially after a bottleneck. Question 2, Gate 2014. Which of the following best describes genetic drift? A. A mechanism of evolution where certain alleles increase in frequency due to environmental factors. B. Random fluctuations in allele frequencies due to chance events. C. The differential survival and reproduction of individuals with certain traits. D. The migration of alleles between populations. Answer. Correct answer is B. Random fluctuations in allele frequencies due to chance events. Genetic drift occurs purely due to chance and is not influenced by the fitness of alleles or their effects on the organism. Summary. Genetic drift is a random process that can cause significant changes in the allele frequencies of a population, especially in small populations. It leads to the loss of genetic variation and can cause alleles to become fixed or lost purely chance. The effects drift are more pronounced in small populations and are responsible for many evolutionary changes that are not driven by chance. Bottleneck and founder effects are two key mechanisms through which genetic drift can occur and have important consequences for the genetic structure of populations. Understanding genetic drift is crucial for studying evolutionary processes and the dynamics of population genetics. UED, Selection ChatGPT said, ChatGPT. Selection, Mechanisms of Evolution. Selection is one of the primary mechanisms of evolution, in which certain traits become more or less common in a population due to the differential reproductive success of individuals possessing those traits. There are different forms of selection, including natural selection, sexual selection, and artificial selection, each of which operates based on different criteria. 1. Natural selection. Natural selection is of individuals with advantageous traits that increase their chances of survival and reproduction are more likely to pass on their genes to the next generation. Over time, this leads to the accumulation of beneficial traits within a population and the elimination of harmful or neutral traits. Key components of natural selection. Variation. Individuals in a population must vary in their traits. These variations arise due to mutations, recombination, and genetic drift. Differential survival. Some individuals are more likely to survive and reproduce due to their advantageous traits. These traits may include better camouflage, faster running speed, or more effective defense mechanisms. Heritability. The traits that confer a survival advantage must be heritable, meaning they can be passed down to offspring. Reproductive success. Organisms with advantageous traits are more likely to reproduce and pass their genes on to the next generation, thereby increasing the frequency of those traits in the population. 2. Types of selection. Selection operates through different modes depending on the effect it has on the phenotypic distribution of traits in a population. These modes of selection include stabilizing selection, directional selection, and disruptive selection. Stabilizing selection. Stabilizing selection occurs when individuals with intermediate traits have higher fitness compared to individuals with extreme traits. It reduces variation. Example, human birth weight. Babies with an intermediate birth weight, not too small or too large, have a higher chance of survival, so these individuals are favored by natural selection. Directional selection. Definition. Direction occurs when one extreme phenotype is favored over others, leading to a shift in the population's trait distribution in one direction. Example, the peppered moth during the Industrial Revolution. Dark-colored moths had higher survival rates due to the pollution-darkened trees, so the population shifted toward darker coloration. Disruptive selection. Disruptive selection occurs when both extreme phenotypes are favored, and intermediate phenotypes are selected against. This can lead to the formation of two distinct groups within the population. Example, a poppy birds with either very small or very large beaks might have an advantage over those with medium-sized beaks if the available food source is either very small seeds or very large seeds. Sexual selection. Sexual selection is a form of natural selection that acts on traits that increase an individual's chances of attracting a mate and reproducing. Unlike natural selection, which is focused on survival, sexual selection focuses on reproductive success. Types of sexual selection. Sexual selection, mate choice, involves one sex, usually females, choosing mates based on certain traits such as bright plumage, impressive displays, or other signs of genetic quality. Sexual selection competition involves competition between members of the same sex, usually males, for access to mates. This could involve physical combat or displays of strength. Example, peafowl peacocks exhibit sexual selection, where females choose mates based on the size and quality of the male's tail feathers. Larger or colorful tails are an indicator of good genes, leading to higher reproductive success. 4. Artificial selection. Artificial selection, also known as selective breeding, is the process by which humans intentionally breed individuals with desirable traits. Unlike natural selection, artificial selection is guided by human choices rather than survival pressures. Example, the domestication of dogs, where humans have selectively bred dogs for specific traits, such as herding ability, size, coat color, or temperament. 5. Key mechanisms of selection. Fitness refers to an individual's ability to survive and reproduce in a specific environment. Individuals with traits that increase their fitness have a higher chance of passing on their genes to the next generation. Adaptation. Adaptation is the process by which a population becomes better suited to its environment due to natural selection acting on beneficial traits. Genetic variation. Selection operates on genetic variation within a population. Genetic variation provides the raw material for selection, allowing populations to adapt to changing environments over time. 6. The role of selection in evolution. Selection is a powerful mechanism driving evolutionary change. Over many generations, selection can lead to the fixation of advantageous traits, the loss of disadvantageous traits, and the formation of new species. Selection is responsible for the adaptation of populations to their environments and can result in the evolution of complex traits such as intelligence, resistance to disease, and specialized behaviors. 7. Practice questions and answers. Question 1, GATE 2015. In a population of butterflies, the distribution of wing color is bimodal. One group of butterflies has white wings and the other group has black wings. If intermediate wing colors are less fit than the extremes, which type of selection is acting on the population? Directional selection B, disruptive selection C, stabilizing selection D, sexual selection. Answer. The correct answer is B, disruptive selection. This selection favors the extremes white and black wings and selects against the intermediate phenotypes, which is consistent with the description. Question 2, GATE 2017. In a population of beetles, individuals with larger body sizes have a higher survival rate due to their ability to resist predators. Which type of selection is most likely to operate in this population? Stabilizing selection B, directional selection C, disruptive selection D, sexual selection. Answer. The correct answer is directional selection. Life favors larger body sizes, causing a shift in the population's trait distribution towards the larger size. Summary. Selection is a key mechanism of evolution that results in changes in the frequency of traits in a population over time. Natural selection favors traits that increase survival and reproductive success, leading to adaptation. Sexual selection acts on traits that influence mate choice or competition for mates, and artificial selection is driven by human and driven. Different types of selection, stabilizing, directional, and disruptive, play distinct roles in shaping the genetic structure of populations. Understanding selection is critical for studying evolutionary processes, as it is a driving force behind the adaptation of species to their environments. You said. One locus two alleles model, ChatGPT said. ChatGPT. One locus two alleles model. One locus two alleles model is a simple framework used in population genetics to describe the genetic variation at a single locus position on a chromosome with two alternative alleles. This model is often used to illustrate fundamental concepts in genetic evolution and the mechanisms affecting allele frequencies within a population. It is crucial in understanding the basic principles of Hardy-Weinberg equilibrium, evolutionary forces like selection, drift, and mutation, and how genetic diversity is maintained or changes over time. One locus two alleles model. In the one locus two alleles model, the following assumptions are typically made. Only two alleles. There are only two alleles at a given genetic locus, typically denoted as A dominant and A recessive. Diploid organisms. Individuals are diploid, meaning they have two copies of each gene, one from each parent. Random mating. Mating is random, meaning that allele combinations, genotypes, are formed by chance. No evolution. Initially, we assume no evolutionary forces are acting on the population, i.e., no selection, drift, or mutation, and allele frequencies are constant over time. 2. Genotype frequencies in the one locus two alleles model. In a population consisting of individuals with two alleles, A and A, individuals can have one of three possible genotypes. A homozygous dominant, A heterozygous, A homozygous recessive, if the frequency of allele A is denoted by P in the frequency of allele A by Q. The allele frequencies must satisfy the equation P plus Q equals 1. P plus Q equals 1 where P equals frequency of allele A. Q equals frequency of allele A. The genotype frequencies in the population can be calculated using Hardy-Weinberg equilibrium principles, which predict the distribution of genotypes under random mating and no evolutionary forces. The expected genotype frequencies are A, P2, P2, A, 2PQ, 2PQ, A, Q2, Q2. 3. Hardy-Weinberg equilibrium in the one locus two alleles model. The Hardy-Weinberg equilibrium describes a condition where allele frequencies remain constant from generation to generation in the absence of evolutionary influences. For a population to be in Hardy-Weinberg equilibrium, the following conditions must be met. Large population size, no genetic drift, random mating, no inbreeding or associative mating, no mutation, no new alleles entering the population, no migration, no gene flow. No natural selection, no differential survival or reproduction. Under these conditions, the allele frequencies P and Q will remain constant, and the genotype frequencies will be in the proportions P 2P2, 2PQ, 2PQ, and Q 2Q2. 4. Evolutionary forces acting on the one locus two alleles model. While the Hardy-Weinberg equilibrium provides a baseline for genetic stability, evolutionary forces can alter allele frequencies over time. These forces include natural selection. If one genotype has a higher fitness, survival, and reproductive success than another, the allele for that genotype will increase in frequency over generation. Example, if allele A confers resistance to disease, individuals with the AA or AA genotype may have a higher survival rate, increasing the frequency of A. Over time, genetic drift. In small populations, allele frequencies can fluctuate randomly over generations due to chance events. This is more pronounced when population sizes are small. Example, if a small population has a random event where all individuals with allele A die, the allele A will become fixed in the population. Mutation. Mutations can introduce new alleles into the population, changing the allele frequencies. Example, a mutation may create a new allele A. Shift allele frequencies from A and A. Gene flow, migration. Immigration or individuals from different populations can introduce new alleles, affecting the allele frequencies in the population. Example, if a population migrates into a region where allele A is common, the allele frequency will shift. 5. Evolutionary change in the one locus two alleles model. If evolutionary forces are acting on the population, changes in allele frequencies can be tracked using the following equations. Change in allele frequency due to selection. If allele A has a selective advantage, its frequency P will increase over time, and Q will decrease. The rate of change in allele frequency can be calculated using the formula, delta. P is dot operator WA minus WA, W delta P equals W. P dot operator WA minus WA, where WA and WA are the fitness values of the genotypes A and A, respectively. W is the mean fitness of the population. Allele frequency due to genetic drift. In small populations, the frequency of an allele can change due to random sampling of alleles. The rate of drift is higher in smaller populations and can be estimated by delta p equals 1 to n dot operator p dot operator 1 minus p delta p equals 2 n1 dot operator p dot operator 1 minus p, where n is the effective population size. Cracked questions and answers. Question 1, GATE 2018. In a population of butterflies, the frequency of allele A is 0.7, and the frequency of allele A is 0.3. The expected frequency of heterozygous individuals, genotype A, under Hardy-Weinberg equilibrium 0.21b, 0.42c, 0.49d, 0.63. Answer. The correct answer is B, 0.42. Using the Hardy-Weinberg equation for heterozygotes, 2 PQ equals 2, 0.7, 0.3 equals 0.42. 2 PQ equals 2, 0.7, 0.3 equals 0.42. Question 2, GATE 2019. In a population, the allele frequencies for a gene are P equals 0.6. P equals 0.6 for allele A. And Q equals 0.4. Q equals 0.4 for allele A. If selection favors a heterozygous genotype, which of the following would you expect? The frequency of allele A will increase. B. The frequency of allele A will increase. C. The frequency of allele A will remain constant. D. The genotype frequencies will shift toward the homozygous genotypes. Answer. The correct answer is that the frequency of allele A will increase. Selection favoring the heterozygous genotype usually results in the balancing of allele frequencies and the increase in frequency of the alleles associated with the heterozygote. Summary. The 1:2 alleles model is a simple yet powerful tool for understanding genetic variation at a single locus. It provides the foundation for more complex population genetic models and helps in understanding how evolutionary forces like selection, drift, mutation, and gene flow influence allele frequencies. The Weinberg equilibrium serves as a benchmark for understanding how populations evolve over time, and deviations from this equilibrium can be attributed to these evolutionary forces. You said. Population genetic structure. ChatGPT said. ChatGPT. Population genetic structure. Population genetic structure refers to the distribution and variation of genetic material across populations. It describes the degree of genetic differentiation between subpopulations within a larger population or between different populations of the same species. The genetic structure of evolutionary forces such as selection, mutation, migration, gene flow, and genetic drift, and it can vary in response to environmental pressures and reproductive strategies. Understanding population genetic structure is key in areas such as evolutionary biology, conservation genetics, and ecology. It allows us to check diversity is maintained, how populations evolve over time, and how they adapt to local conditions. Key concepts in population genetic structure. Population, a group of interbreeding individuals of the same species, living in the same area at the same time, with the potential to exchange genes. Gene pool, the complete set of alleles present in a population, which includes all the genetic variation within that group. Variation, differences in DNA sequences between individuals in a population, which may lead to variations in traits phenotypes. Subpopulations, smaller groups within a population that have some degree of genetic differentiation due to factors like geographic isolation or behavioral differences. Factors influencing population genetic structure. Gene flow, migration. Gene flow refers to the movement of genes or alleles between populations due to migration or dispersal. When individuals from different populations interbreed, they introduce new genetic material into the gene pool, reducing genetic differentiation between populations. High flow tends to reduce genetic differentiation, whereas low gene flow allows for more local adaptation and increased genetic differences between populations. Migration of individuals from one population of birds to another population can introduce new alleles, reducing genetic differences between the populations. Genetic drift. Genetic drift is the random fluctuation of allele frequencies due to chance events, especially in small populations. Over time, genetic drift can lead to the loss or fixation of alleles and can result in significant genetic differentiation between populations. A drastic reduction in population size due to events like natural disasters can cause a loss of genetic diversity. Founder effect. When a small group of individuals forms a new population, the genetic makeup of the new population may differ significantly from the original population due to the limited genetic diversity of the found. Natural selection. Natural selection can shape the genetic structure of a population by favoring certain alleles over others based on environmental pressures. If environments favor different traits, populations in these environments may evolve distinct genetic structures. When populations experience different environmental conditions, they may adapt in unique ways, leading to genetic differentiation between populations. Mutation. Mutation introduces new genetic variation in population by altering the DNA sequence. Although most mutations are neutral or harmful, beneficial mutations can become more common due to selection. Over time, mutations contribute to the differentiation of populations, especially in isolated populations where gene flow is limited. Isolation. Geographic isolation, e.g., mountains, rivers, or ecological isolation, e.g., different niches can prevent gene flow between populations, leading to genetic divergence over time. Isolation is a key factor in the formation of new species, as it reduces the chances of interbreeding and allows genetic differences to accumulate. Measuring population genetic structure. F statistics fixation index. F statistics are used to quantify the genetic structure of populations and measure the degree of inbreeding or genetic differentiation between subpopulations. F_is measures the level of inbreeding within subpopulations, individuals relative to their subpopulation. F_ST measures the degree of genetic differentiation between subpopulations. I_ST value indicates that subpopulations are genetically distinct, while a low value suggests that they share a high degree of genetic similarity. Heterozygosity. Heterozygosity refers to the proportion of individuals in a population that are heterozygous for a given locus carry two different alleles. Heterozygosity is typically associated with a large, genetically diverse population. Decrease in heterozygosity may indicate inbreeding, genetic drift, or reduced gene flow. Genetic distance. Genetic distance measures the genetic divergence between two populations. It is often calculated based on allele frequencies or molecular markers such as microsatellites or DNA sequences. Genetic distance equals 1, similarity coefficient. AMOVA, analysis of molecular variance. AMOVA is a statistical method used to analyze genetic variation within and among populations. It partitions the total genetic variation into components corresponding to differences within populations, among populations within groups, and among groups. It helps determine whether genetic differences are primarily due to differences between groups or if they are distributed within populations. Models of population genetic structure. Island model. In the island model, populations are structured as a set of islands, each with its own gene pool. Gene flow occurs between neighboring islands, and individuals can migrate between them. It assumes that the populations are genetically similar but may develop differentiation over time due to isolation by distance or local adaptation. Isolation by distance model. This model assumes that populations that are geographically closer are more genetically similar due to higher rates of gene flow between them. Between populations increases, gene flow decreases, and genetic differentiation increases. It is used to explain patterns of genetic variation observed in natural populations that are not fully isolated but experience limited gene flow. Stepping stone model. This model is similar to the island model but assumes that migration occurs only between neighboring populations in a linear or grid-like arrangement. This structure may result in a pattern of genetic differentiation that reflects the distances between populations. Importance of studying population genetic structure. Conservation genetics. Understanding the genetic structure of endangered species or populations allows for better management strategies. Populations with high genetic differentiation may require separate conservation efforts to preserve their unique genetic diversity. In the conservation of the cheetah, understanding the genetic structure of different populations can help in deciding whether to establish corridors for gene flow or to manage them as distinct populations. Evolutionary biology. Population genetic structure provides insight into the evolutionary processes that shape genetic diversity and adaptation. Studying and how genetic differentiation arises can shed light on the mechanisms of speciation and evolutionary dynamics. Example, the genetic structure of Darwin's finches on the Galapagos Islands has provided insights into how different populations adapt to varying ecological niches. Human genetics. The study of human population genetic structure helps in understanding the patterns of migration, ancestry, and genetic disease distribution. It has implications for understanding the spread of diseases, genetic predispositions, and human evolution. Genetic variation in human populations has been instrumental in studying the origins and migration of early human ancestors. Practice questions and answers. Question 1, Gate 2017. Which of the following is the primary factor that maintains the genetic structure of a population over time? A. Genetic drift. B. Natural selection. C. Gene flow. D. Mutation. Answer. The correct answer is C. Gene flow. Gene flow is the product that produces new alleles into a population, thus helping to maintain or alter its genetic structure over time. It acts to reduce genetic differentiation between populations. Question 2, Gate 2020. In a population with two subpopulations, the genetic differentiation between the subpopulations is measured by F_ST. If F_ST is close to 1, what does this indicate? High differentiation between the subpopulations. B. Low genetic differentiation between the subpopulations. C. No genetic variation in the population. D. Equal allele frequencies in the subpopulations. Answer. The correct answer is a high genetic differentiation between the subpopulations. An F value close to 1 indicates that the genetic differentiation between the subpopulations is high, meaning that the populations are genetically distinct from one another. Summary. Population genetic structure is a critical aspect of evolutionary biology that reflects the distribution of genetic variation within and between populations. Evolutionary forces, such as gene flow, genetic drift, natural selection, and mutation, play a role in shaping the genetic structure of populations. By studying genetic structure, scientists can gain insights into evolutionary processes, biodiversity, and the management of species, especially in conservation contexts. Panmixia, ChatGPT said. ChatGPT Panmixia. Refers to a situation in which all individuals in a population are equally likely to mate with each other, regardless of their genetic makeup, age, sex, or other characteristics. It's a condition where there is random mating, and no particular selection or geographic or behavioral isolation occurs. Exhibiting panmixia, all individuals have an equal chance of contributing their genes to the next generation, resulting in no reproductive barriers. In a mixed population, allele frequencies are expected to remain stable over generations, assuming that no evolutionary forces, such as selection, migration, or genetic drift, act on the population. Equilibrium is sometimes referred to as Hardy-Weinberg equilibrium, which assumes panmixia along with other conditions such as no selection or mutation. Key features of panmixia: random mating. Every individual in the population has an equal probability of mating with any other individual, leading to a random mixing of genes. No geographic or behavioral isolation. Panmixed population, there are no barriers, geographical or behavioral, that prevent gene flow between individuals. All members are considered genetically equally available. Equal reproductive success. There is no selective pressure on specific phenotypes or genotypes. This means that all individuals, regardless of their characteristics, are equally likely to reproduce and pass on their genes to the next generation. Random genetic drift. While Panmixia assumes random mating, genetic drift can still play a role in small populations, leading to the random fluctuation of allele frequencies. However, in large populations, drift may have minimal effects compared to natural selection or gene flow. No speciation or subpopulation structure. In Panmixia, there is no divergence into different subpopulations with distinct genetic compositions. The population as a whole shares a gene pool with little or no genetic differentiation across the population. Factors impacting Panmixia. Although assumes random mating, in reality, factors can limit or break Panmixia in natural populations. Sexual selection. Are favored by mates, e.g., bright coloration or elaborate courtship. Individuals with these traits may have more reproductive success than others, leading to non-random mating and a departure from Panmixia. Geographic or ecological barriers. Barriers such as mountains, rivers, or oceans can lead to geographic isolation, reducing gene flow between subpopulations and preventing Panmixia. Behavioral isolation. In mating behaviors, e.g., timing of mating seasons, mating rituals, or habitat preferences can result in some individuals not having the opportunity to mate with others, thus disrupting Panmixia. Inbreeding. In small populations, inbreeding can occur, where individuals tend to mate with close relatives rather than randomly with all available mates. This reduces genetic diversity and is the opposite of Panmixia. Dissociative mating. Net members based on specific characteristics, e.g., mating with genetically dissimilar individuals will violate Panmixia. Panmixia in evolutionary theory. Hardy-Weinberg equilibrium. Hardy-Weinberg principle assumes that a population is panmictic and that there are no evolutionary forces acting on it, i.e., no selection, migration, mutation, or genetic drift. Under these conditions, allele frequencies in the population will remain constant from generation to generation. The Hardy-Weinberg equation is given by P 2 plus 2pq plus q2 equals 1p2 plus 2pq plus q2 equals 1 where p2 is the frequency of the homozygous dominant genotype. 2 pq 2pq is the frequency of the heterozygous genotype. q 2q2 is the frequency of the homozygous recessive genotype. Effect of Panmixia on genetic diversity. In a large, panmictic population, genetic diversity tends to remain stable over time, as there are no barriers to gene flow. But factors such as genetic drift, mutation, and natural selection can still influence genetic variation. Role in speciation. Panmixia may prevent speciation in populations, as gene flow is uninterrupted. On the other hand, of isolation or disruption in Panmixia, such as allopatric isolation, can lead to divergence in speciation. Applications of Panmixia in population genetics. Assumption and population models. Many population genetic models, such as those used to calculate allele frequencies under Hardy-Weinberg equilibrium, assume Panmixia. These models provide a baseline for comparing real populations and understanding how evolutionary forces like natural selection or migration alter genetic structures. Conservation genetics. It can be important in conservation biology, as maintaining high levels of gene flow within populations can prevent inbreeding depression and ensure genetic diversity, which is crucial for long-term survival and adaptation. Human populations. In human populations, Panmixia is rarely fully realized due to social, cultural, and geographical factors. However, understanding the concept can help explain how populations intermingle and share genetic material over time, such as in large urban centers with diverse populations. Practice questions and answers. Question 1, Gate 2019. Which of the following conditions would violate the assumption of Panmixia in a population? Equal opportunity for mating among all individuals. B. Random mating within the population. C. Non-random mating based on phenotype. D. No selective pressures acting on the population. Answer. The correct answer is C. Non-random mating based on phenotype. Non-random mating selection where individuals choose mates based on specific traits violates the assumption of Panmixia, where all individuals should have an equal chance of mating. Question 2, Gate 2021. In a population that is in Hardy-Weinberg equilibrium, which of the following is not assumed? Random mating, Panmixia. B. No genetic drift. C. No gene flow. C. Natural selection is acting on the population. Answer. The correct answer is C. Natural selection is acting on the population. The Hardy-Weinberg equilibrium assumes that no natural selection, mutation, or gene flow occurs, and that there is random mating Panmixia. Natural selection would disrupt Hardy-Weinberg equilibrium by favoring specific alleles. Summary. Panmixia represents a theoretical model where all individuals in a population mate randomly, without preference for specific traits or genetic backgrounds. This is key to the Hardy-Weinberg equilibrium, where allele frequencies remain stable over generations in the absence of evolutionary forces. Often an assumption in population genetics, real-world populations rarely meet this condition due to factors such as natural selection, genetic drift, and geographic or behavioral isolation. Understanding Panmixia provides insights into genetic diversity, gene flow, and evolutionary dynamics in populations. Gene flow, ChatGPT said. ChatGPT gene flow. Gene flow, also known as gene migration or allele flow, is the transfer of genetic material between distinct populations. Individuals from different populations interbreed, introducing new alleles into a population's gene pool. Gene flow is one of the key mechanisms of evolution, alongside other factors like natural selection, mutation, and genetic drift. It plays a crucial role in maintaining genetic diversity within populations and preventing speciation. Key features of gene flow: movement of alleles. Gene flow involves the movement of alleles between populations through migration, which can either increase or decrease genetic variation in the receiving population. Homogenizing effect. The most significant impacts of gene flow is its tendency to reduce genetic differentiation between populations. By introducing new alleles into a population, gene flow can make genetic variation more similar across populations. Cross-population breeding. For gene flow to occur, individuals from different populations must breed. This may involve the migration of individual organisms, such as animals or plants, from one population to another. Can prevent speciation. Gene flow can prevent populations from becoming reproductively isolated and evolving into distinct species. It acts as a homogenizing force, preventing populations from diverging genetically. Mechanisms of gene flow. Migration. The movement of individuals or their gametes from one population to another is the most direct mechanism of gene flow. Migration is often seasonal, driven by factors such as food availability, mating behaviors, or environmental changes. Dispersal. Dispersal is the movement of individuals or seeds in plants away from their birthplace to establish a new colony. Successful dispersal can introduce genetic material into new populations, resulting in gene flow. Pollination in plants. For plants, gene flow often occurs pollination. Pollen can be carried by wind, water, or animals to other plants, facilitating gene flow between different populations of the same species. Hybridization. When two distinct populations come into contact, hybridization can occur if they are closely related. Hybrid offspring can contribute alleles to the gene pool of both parental populations, promoting gene flow. Factors affecting gene flow. Geographic barriers. Physical barriers like mountains, oceans, rivers, or deserts can prevent gene flow by isolating populations. In such cases, even if the populations are genetically compatible, they may not have the opportunity to interbreed. Behavioral barriers. Behavioral differences, such as mating rituals or preferences, can limit gene flow. For example, two populations of animals may be capable of interbreeding but may have different mating behaviors that prevent successful reproduction. Temporal isolation. When two populations reproduce at different times, e.g., different breeding seasons, gene flow is restricted. This is often seen in plants that flower at different times or animals that mate in different seasons. Genetic compatibility. Even if gene flow occurs between populations, if the populations are genetically too different, successful reproduction may not occur. Hybridability or inviability is an example of such incompatibility, as seen in many interspecies hybrid crosses, e.g., mules. Human activities. Human activities like agriculture, urbanization, and the introduction of invasive species have drastically altered patterns of gene flow. For instance, human-mediated transportation can introduce species to new habitats, facilitating gene flow between formerly isolated populations. Role of gene flow in evolution. Increasing genetic diversity. By introducing alleles into a population, gene flow increases genetic diversity, which can provide a population with more raw material for natural selection to act upon. Counteracting the effects of genetic drift. Gene flow can counteract the loss of genetic diversity due to genetic drift in small populations. By consciously introducing new alleles, gene flow maintains the adaptive potential of a population. Prevents divergence and speciation. Gene flow reduces the likelihood of speciation by maintaining genetic connectivity between populations. When gene flow is significant, it prevents populations from becoming genetically isolated and evolving independently into separate species. Increases fitness in new environments. When individuals migrate to a new environment, they may bring genetic adaptations that enhance the fitness of the receiving population in that environment. This process, often referred to as adaptive gene flow, can help populations adapt to changing conditions. Gene flow and speciation. Gene flow is a barrier to speciation. In populations where gene flow is high, populations tend to remain genetically similar, and speciation is less likely to occur. Speciation usually happens when gene flow is restricted, leading to reproductive isolation and genetic divergence. Allopatric speciation. In allopatric speciation, populations become geographically isolated, preventing gene flow between them. Over time, these populations accumulate genetic differences due to mutation, genetic drift, and selection, leading to speciation. Sympatric speciation. In sympatric speciation, populations are not geographically isolated, but behavioral, ecological, or temporal factors may limit gene flow within the population. Over time, these factors may lead to reproductive isolation and speciation, even without physical barriers. Parapatric speciation. Parapatric speciation occurs when populations are partially isolated by a geographic or environmental barrier, but there is still some gene flow. The reduced gene flow and selection pressures in different environments may lead to speciation over time. Applications of gene flow. Conservation biology. Gene flow is essential for maintaining genetic diversity within endangered species. Ensuring that isolated populations experience gene flow can prevent inbreeding depression and increase the chances of species survival. Agriculture and breeding. In agricultural crops and domesticated animals, gene flow is manipulated through selective breeding to improve desirable traits. Controlled gene flow is often used to introduce beneficial traits such as disease resistance, drought tolerance, or higher yields. Invasive species. Gene flow can play a role in the success of invasive species. The introduction of new genetic material through hybridization with native species can lead to increased fitness, helping invasive species adapt more quickly to new environments. Disease ecology. Understanding gene flow in pathogen populations, such as viruses or bacteria, is crucial for tracking the spread of diseases. Changes in pathogen gene flow patterns can affect the evolution of virulence, resistance to treatments, and the spread of infections. Practice questions and answers. Question 1, Gate 2017. Which of the following would not be a consequence of gene flow in a population? A. Increased gene diversity. B. Decreased allele frequency differences between populations. C. Increased potential for adaptation to new environments. D. Reduced mutation rate. Answer. The correct answer is D. Reduced mutation rate. Gene flow does not directly affect the mutation rate. Instead, it can introduce new alleles and increase genetic diversity. The other options are Which of the following would not be a consequence of gene flow in a population? Which of the following would not be a consequence of gene flow in a population? A. Increased genetic diversity. B. Decreased allele frequency differences between populations. C. Increased potential for adaptation to new environments. D. Reduced mutation rate. Answer. The correct answer is D. Reduced mutation rate. Gene flow does not directly affect the mutation rate. Instead, it can introduce new alleles and increase genetic diversity. The other options are all consequences of gene flow in populations. Question 2, Gate 2020. Gene flow tends to counteract which of the following evolutionary processes? A. Genetic drift B. Natural selection C. Mutation D. Both B and C. Answer. The correct answer is a genetic drift. Gene flow can counteract genetic drift by introducing new alleles into a population, thus maintaining genetic variation and preventing random fluctuations in allele frequencies, especially in small populations. Summary. Gene flow is an evolutionary force that involves the movement of alleles between populations, leading to increased genetic diversity and preventing speciation. It occurs through mechanisms such as migration, dispersal, hybridization, and pollination. Although gene flow tends to reduce genetic differentiation and promotes the mixing of genes, it can be hindered by geographic or behavioral barriers. In conservation, agriculture, and disease management, understanding gene flow is essential for maintaining genetic health, adapting to new environments, and preventing the spread of harmful traits. You said, FST. ChatGPT said, ChatGPT. FST Fixation Index. FST is a measure of population differentiation based on genetic data, particularly used to quantify the amount of genetic variation that occurs between populations relative to the total genetic variation in a species or group. It is widely employed in population genetics to study the level of genetic differentiation between subpopulations and to understand the mechanisms of evolution such as gene flow, genetic drift, and natural selection. Key concepts of FST definition. FST represents the fraction of genetic variance that is due to differences between populations. It can be seen as a measure of population differentiation and is often used to infer the extent of genetic isolation between populations. Mathematical formula. FST is calculated using the formula F. ST equals VBVT FST equals VT. VB. Where VB is the genetic variance between populations. VT is the total genetic variance which includes both within population and between population variants. Alternatively, FST can be expressed as FST equals HT minus HS HT FST equals HT. HT minus HS. Where HT is the expected heterozygosity at the total population level across all subpopulations. SHS is the expected heterozygosity within subpopulations. Interpretation of FST values. FST equals 0. No genetic differentiation between populations. All the genetic variation is found within populations. FST equals 1. Complete genetic differentiation. All the genetic variation is found between populations, with no shared alleles. 0 in LT, FST and LT. 1. A positive value indicates some degree of genetic differentiation between populations, with the value increasing as the degree of differentiation rises. A higher FST value generally indicates lower gene flow or higher genetic differentiation between populations. Implications of FST. Low FST and LT, 0.05 indicates that populations are genetically similar, suggesting high gene flow between populations and or recent divergence. Moderate FST, 0.05 minus 0.15, suggests moderate differentiation, possibly due to limited gene flow or the influence of genetic drift. High FST and GT, 0.15, indicates significant genetic differentiation, possibly due to restricted gene flow, geographic barriers, or strong selection pressures. Factors affecting FST. Gene flow. High gene flow between populations reduces genetic differentiation, leading to lower FST values. Conversely, low gene flow leads to higher FST, as populations become genetically isolated. Selection. Natural selection can drive divergence between populations, particularly if different selective pressures exist in different environments. This can result in higher FST values. Genetic drift. In small populations, genetic drift can cause random changes in allele frequencies, which can increase FST. Over time, small isolated populations may develop unique genetic profiles due to drift. Population size. The size of populations can influence FST. Larger populations typically have greater genetic diversity, which can lower FST when compared to smaller populations, where drift can play a larger role. Geographic isolation. Geographic or physical barriers, e.g., mountains, rivers, deserts, can limit gene flow between populations, increasing genetic differentiation and, thus, increasing FST. Applications of FST. Measuring genetic differentiation. FST is primarily used to assess genetic differentiation between populations. Comparing FST values between different groups, researchers can determine how isolated populations are from each other and how much gene flow occurs between them, inferring the role of evolutionary processes. High FST values suggest limited gene flow and possible geographic or ecological isolation, while low FST values point to strong gene flow and potential recent interbreeding between populations. This can be used to infer past evolutionary events. Conservation genetics. FST is crucial in conservation biology. It helps identify populations that are genetically isolated and may require special conservation efforts to prevent inbreeding depression or loss of genetic diversity. Populations with high FST may be at risk of losing genetic diversity due to restricted gene flow. Speciation studies. FST is often used to study speciation, especially in cases of allopatric speciation, where populations are geographically isolated. Over time, these isolated populations accumulate genetic differences, leading to high FST values and eventually, reproductive isolation. Human genetics and disease studies. FST can be used in human genetics to study the genetic structure of human populations. It helps in understanding the distribution of genetic diseases across different populations and how different subpopulations may have evolved distinct genetic traits or resistances to certain diseases. Calculation of FST. FST can be calculated using a variety of approaches depending on the type of genetic data available. Some common methods include microsatellites. Microsatellite data is commonly used to calculate FST as it provides high-resolution genetic markers. Microsatellites are highly variable and can detect small genetic differences between populations. Single nucleotide polymorphisms, SNPs. SNP data is increasingly used to calculate FST, especially with high-density genotyping techniques. SNPs are less variable than microsatellites but are more abundant and are widely used in large-scale population genetics studies. Mitochondrial DNA are nuclear markers. In some cases, FST is calculated using mitochondrial DNA, mtDNA, or nuclear DNA markers. mtDNA is often used in studies of maternal lineages, while nuclear markers can give a broader view of genetic differentiation. Practice questions and answers. Question 1, GATE 2016. Which of the following statements about FST is true? A. FST values are always between 0 and 1. B. FST represents the proportion of genetic variation within populations. C. A high FST value suggests high gene flow between populations. D. A low FST value suggests that populations are genetically isolated. Answer. The correct answer is that FST values are always between 0 and 1. FST values range from 0 to 1, with 0 indicating no genetic differentiation between populations and 1 indicating complete differentiation. Question 2, GATE 2018. An FST value of 0.25 indicates that the populations under study are A. Genetically identical. B. Highly genetically differentiated. C. Moderately genetically differentiated. D. Incomplete samples. Answer. The correct answer is C. Moderately genetically differentiated. An FST value of 0.25 indicates moderate genetic differentiation, suggesting that there is some degree of isolation or reduced gene flow between populations. Summary. FST is a fundamental measure used in population genetics to understand the genetic structure of populations and the degree of differentiation between them. It is widely used in studies of evolutionary processes, such as gene flow, genetic drift, and natural selection. By assessing FST, scientists can gain insight into the historical and ongoing forces shaping the genetic diversity within and between populations. FST has applications in conservation, speciation studies, and disease ecology, making it an essential tool for understanding the genetic dynamics of populations. You said, polygenic traits. ChatGPT said, ChatGPT. Polygenic traits. Polygenic traits refer to characteristics that are influenced by multiple genes, each contributing a small amount to the overall phenotype. Unlike Mendelian traits, which are controlled by a single gene, polygenic traits are the result of the interaction between several genes, each located at different loci on the chromosomes. These traits are often continuously distributed in the population, exhibiting a range of phenotypes. Key characteristics of polygenic traits: multifactorial inheritance. Polygenic traits are typically controlled by 2 or more genes, often with environmental factors also playing a role in determining the phenotype. These traits are often complex and are the result of the combined effect of the alleles at multiple loci. Continuous distribution. One of the hallmarks of polygenic traits is their continuous distribution. Unlike traits controlled by a single gene, which typically show discrete categories, e.g., tall versus short in Mendelian inheritance, polygenic traits show a range of phenotypes. Examples include human height, skin color, and eye color, which do not exhibit a clear-cut classification but rather a spectrum. Additive effect of alleles. In polygenic traits, the individual alleles contribute additively to the phenotype. For example, each allele might contribute a small increment in height or skin pigmentation, and the overall phenotype results from the accumulation of these small effects. No clear dominance or recessiveness. In contrast to Mendelian traits where dominant and recessive alleles influence the phenotype, polygenic traits often lack such clear dominance or recessiveness. The alleles at multiple loci may all contribute equally to the phenotype or have varying levels of contribution, but there is no simple dominant recessive pattern. Examples of polygenic traits. Human height. Human height is a classic example of a polygenic trait. It is influenced by multiple genes, possibly hundreds, spread across the genome. These work in conjunction with environmental factors like nutrition and health during childhood to determine an individual's height. Skin color. Skin color in humans is also a polygenic trait. It is influenced by the interactions of several genes responsible for the production of melanin, the pigment that gives skin its color. The variation in the amount and type of melanin produced results in a continuous spectrum of skin tones. Eye color. Although once thought to follow a simple Mendelian inheritance pattern, eye color is now known to be influenced by multiple genes, with each gene contributing to the overall pigmentation. Intelligence. Intelligence is another complex trait that is influenced by many genes, along with environmental factors. It indicates that there is a polygenic component to intelligence, where multiple genetic variations contribute to cognitive ability. Diseases. Some diseases, such as diabetes, heart disease, and certain cancers, are influenced by polygenic inheritance. They may not be caused by a single gene. Multiple genes interact to increase susceptibility to these diseases. Genetic mechanism of polygenic traits. Additive genetic effects. For polygenic traits, the effect of individual alleles is usually additive. For example, if 3 genes each contribute 1 unit to a trait like height, an individual with 3 tall alleles would have a total additive score of 3 units, resulting in a taller phenotype. Interaction between genes. In some cases, genes involved in polygenic traits may interact with each other, resulting in epistasis gene-gene interactions. This means that the effect of one gene may depend on the presence or absence of other genes. For example, one gene may modify or enhance the expression of another gene that influences a trait. Environmental influence. Polygenic traits are often influenced by environmental factors, making them multifactorial. For example, the expression of height may be influenced by nutritional intake during childhood, and the expression of skin color may be affected by exposure to sunlight. Inheritance of polygenic traits. Quantitative inheritance. Polygenic traits follow a pattern of quantitative inheritance where the phenotypic variation is due to the combined effect of multiple genes. This variation is often continuous and measured on a spectrum. Phenotypic distribution. The phenotypic distribution of polygenic traits typically follows a bell-shaped curve, normal distribution, with most individuals clustering around the average phenotype and fewer individuals exhibiting extreme phenotypes. For example, most individuals have a medium height, with fewer individuals being very tall or very short. Segregation ratios. Polygenic traits do not follow simple Mendelian ratios. Instead of observing the typical 3:1 or 1:2:1 ratio seen in Mendelian inheritance, polygenic traits typically show a large number of different phenotypes, with intermediate phenotypes being the most common. For example, if height were controlled by 3 genes, a cross between 2 individuals of extreme heights would produce offspring with a range of intermediate heights. Heritability. Heritability of polygenic traits refers to the proportion of variation in a trait that can be attributed to genetic factors. High heritability suggests that genetic factors play a major role in determining the phenotype, although environmental factors still contribute. Calculation and measurement of polygenic traits. Genetic models. To study polygenic traits, geneticists use polygenic models which assume that the trait is influenced by multiple loci, each contributing a small effect. These models allow researchers to predict the inheritance patterns of polygenic traits in populations. Quantitative trait loci (QTL) mapping. QTL mapping is a technique used to identify the specific regions of the genome that contribute to the variation in a polygenic trait. By analyzing the relationship between genetic markers and phenotypic variation, researchers can pinpoint the loci associated with a trait. Heritability estimates. Heritability can be estimated by examining the relative contribution of genetic and environmental factors to the total phenotypic variation. This is often done using twin studies, family studies, or twin adoption studies, where the genetic similarity between individuals is compared to their phenotypic similarity. Applications of polygenic traits. Selective breeding. In agriculture and animal breeding, polygenic traits are important for the selection of desirable traits, such as larger body size in livestock or improved yield in crops. By selecting individuals with favorable polygenic combinations, breeders can increase the frequency of beneficial alleles in the population. Medical genetics. Understanding polygenic traits is crucial in medical genetics, particularly in the study of diseases with complex inheritance patterns, such as cardiovascular disease, diabetes, and mental health disorders. Identifying the genetic components of these diseases can lead to more personalized approaches to prevention and treatment. Forensics and evolutionary biology. Polygenic traits are important in forensics for the identification of individuals based on genetic profiles. Additionally, studying polygenic traits in evolutionary biology helps explain how traits like skin color, height, and intelligence evolve and adapt to environmental conditions. Practice questions and answers. Question 1, GATE 2019. Which of the following is most characteristic of polygenic inheritance? A. The inheritance of traits follows Mendel's laws. B. The trait exhibits continuous variation in a population. C. Only one gene determines the phenotype. D. There are two possible phenotypes for the trait. Answer. The correct answer is B. The trait exhibits continuous variation in a population. Polygenic traits show a continuous range of phenotypes in a population, as they are influenced by multiple genes. Question 2, GATE 2021. Human height is an example of a polygenic trait. Which of the following best describes its inheritance pattern? A. It follows a Mendelian inheritance pattern. B. It is controlled by a single gene with dominant and recessive alleles. C. It is influenced by multiple genes, each contributing a small effect. D. It is determined solely by environmental factors. Answer. The correct answer is C. It is influenced by multiple genes, each contributing a small effect. Human height is influenced by the additive effect of many genes, and environmental factors like nutrition also contribute to the phenotype. Summary. Polygenic traits are characteristics controlled by multiple genes, each contributing a small amount to the overall phenotype. These traits exhibit continuous variation and do not follow simple Mendelian inheritance patterns. Instead, they are influenced by the combined effect of many alleles, environmental factors, and gene-gene interactions. Examples of polygenic traits include human height, skin color, and intelligence. Understanding polygenic inheritance is crucial for fields such as genetics, medicine, and evolutionary biology. You said. Gene-environment interactions. ChatGPT said. ChatGPT. Gene-environment interactions. Gene-environment interactions refer to the complex interplay between genetic factors and environmental influences that together shape an organism's phenotype. These interactions suggest that the effect of a gene on a particular trait is dependent on the environmental context in which it is expressed. Similarly, the environment can also influence the way in which genetic traits are manifested. While genetics determine the potential for a particular phenotype, the environment can modify how this potential is expressed. In some cases, environmental factors may enhance or suppress the genetic potential, leading to a wide variety of phenotypic outcomes. Key concepts in gene-environment interactions. Nature versus nurture debate. Gene-environment interactions are central to the nature versus nurture debate, where nature refers to genetic inheritance and nurture refers to environmental influences. Modern understanding recognizes that both genetic and environmental factors contribute significantly to the phenotype. Interaction types. Additive effects. In some cases, the effects of genetic and environmental factors are additive, meaning their influence on the phenotype is simply the sum of their independent effects. Epistasis. Genetic interactions between alleles. Epistasis may be influenced by the environment, affecting how traits are expressed. Gene-environment modulation. The environment can modify the expression of genes, especially in polygenic traits, where environmental factors e.g., nutrition, exposure to toxins can influence the final phenotype. Examples of gene-environment interactions. Human height. Genetics largely determines a person's height, but environmental factors such as nutrition during childhood and adolescence play a significant role. For example, a child may inherit a genetic predisposition for tallness, but if they do not have access to proper nutrition, they may not reach their potential height. Skin color. Skin color is influenced by genetic factors, with multiple genes contributing to the amount and type of melanin produced in the skin. However, environmental factors like sun exposure can affect skin pigmentation. People who inherit genes for darker skin color but live in areas with little sun exposure may have lighter skin than they would have if they had lived in sunnier environments. Disease susceptibility, e.g., cancer, heart disease. Diseases, such as cardiovascular disease and cancer, are influenced by a combination of genetic predisposition and environmental risk factors. For instance, an individual with a genetic susceptibility to heart disease may not develop the condition unless they are exposed to environmental risk factors such as poor diet, lack of exercise, or smoking. Mental health disorders. Conditions such as schizophrenia or depression involve both genetic factors and environmental influences. A person may inherit genes that increase the risk of these disorders, but the manifestation of the disorder may depend on environmental triggers, such as stress, trauma, or substance abuse. Drug metabolism. The metabolism of drugs is influenced by genetic variation in enzymes responsible for breaking down medications, e.g., cytochrome P450 enzymes. However, environmental factors, such as diet, other medications, and even gut microbiota, can alter the activity of these enzymes and modify drug efficacy and toxicity. Mechanisms of gene-environment interactions. Gene regulation. Environmental factors can affect gene regulation, including the activation or repression of certain genes. For example, exposure to toxins may turn on genes associated with detoxification, or sunlight may activate genes involved in vitamin D synthesis. Epigenetic modifications. Epigenetics refers to changes in gene expression that do not involve alterations to the DNA sequence itself. Environmental factors, such as diet, stress, and toxins, can lead to epigenetic modifications such as DNA methylation or histone modification, which can influence gene expression and thus phenotype without changing the underlying genetic code. Gene-environment correlation. A gene-environment correlation occurs when genetic predispositions influence an individual's exposure to particular environments. For example, a person with a genetic predisposition for higher intelligence might actively seek out enriching educational experiences, which in turn reinforce their cognitive abilities. Environmental modulation of genetic expression. Some genes may only be expressed in specific environmental conditions. For example, certain species may only express particular characteristics, e.g., flowering time or fruit color, when exposed to particular environmental cues like temperature or light. Types of gene-environment interactions. Additive interaction. This is where the effects of genes in the environment are independent of one another and add up. For instance, a person's weight may be influenced by both genetic predisposition, in terms of metabolic rate, and environmental factors, diet and physical activity. Interactive or non-additive interaction. In these cases, the effect of one factor, gene or environment, is dependent on the presence of the other. For instance, the impact of a stress-related gene might only manifest in the presence of high stress. In the absence of stress, the gene may have no effect on the individual's mental health. Threshold model. This model suggests that environmental factors may influence whether an individual crosses a certain threshold to express a certain phenotype. For example, an individual may have a genetic susceptibility to developing a disease, but they may only manifest the disease if they are exposed to a certain environmental factor, e.g., smoking or a high-fat diet. Methods to study gene-environment interactions. Twin studies. Twin studies are used to estimate the contribution of genetic and environmental factors to a particular trait. Comparing the similarity between monozygotic identical twins who share 100% of their genes and dizygotic fraternal twins who share 50% of their genes, researchers can estimate the relative contributions of genetics and the environment to trait variation. Genome-wide association studies, GWAS. GWAS studies can identify genetic variants associated with diseases or traits and can be used to investigate gene-environment interactions. By correlating genetic data with environmental data, researchers can pinpoint specific environmental factors that interact with genetic variants to influence traits. Animal models. Animal models, e.g., mice or fruit flies, are frequently used to study gene-environment interactions in controlled environments. These models allow researchers to manipulate both genetic makeup and environmental conditions to observe how different factors contribute to phenotypic variation. Longitudinal studies. Longitudinal studies track individuals over time and can help establish how environmental factors interact with genetic predispositions to influence traits and diseases. These studies often include assessments of both genetic data and environmental exposures, e.g., lifestyle, diet, stress, across a person's lifespan. Applications of gene-environment interactions. Personalized medicine. By understanding gene-environment interactions, healthcare providers can offer more personalized treatments based on an individual's genetic makeup and environmental exposures. For example, a person with a genetic predisposition for a disease might be advised to avoid certain environmental triggers, e.g., high-fat diet for heart disease, to reduce the risk of developing the condition. Public health strategies. Knowledge of gene-environment interactions can inform public health strategies aimed at reducing disease risk. For example, reducing exposure to environmental factors, e.g., pollution, smoking may be especially beneficial for individuals with genetic susceptibility to respiratory diseases. Conservation biology. Gene-environment interactions play a role in the adaptation and survival of species in changing environments. Understanding how environmental factors influence the expression of genetic traits in wildlife can help guide conservation efforts, especially in the face of climate change. Practice questions and answers. Question 1, GATE 2020. Which of the following best describes gene-environment interactions? A. Gene-environment interactions occur only in the presence of harmful environmental factors. B. Environmental factors can modify the expression of genes and vice versa. C. Environmental factors do not influence gene expression. D. Genetic factors alone determine an organism's phenotype. Answer. The correct answer is B. Environmental factors can modify the expression of genes and vice versa. Gene-environment interactions involve the influence of both genetic factors and environmental exposures on the phenotype. Question 2, GATE 2022. Which of the following is an example of a gene-environment interaction? A disease that only occurs in individuals with a specific genetic mutation and no environmental influence. B. A plant species that grows taller in nutrient-rich soil compared to nutrient-poor soil, with genetic variation contributing to this difference. C. A disease that manifests in individuals only after exposure to a specific environmental factor, such as stress. D. A trait controlled entirely by genetic factors, unaffected by environmental influence. Answer. Correct answer is C. A disease that manifests in individuals only after exposure to a specific environmental factor, such as stress. This scenario describes a gene-environment interaction where the environment, stress, interacts with genetic susceptibility to cause the disease. Summary. Gene-environment interactions are critical in shaping the phenotype of organisms. These interactions show that both genetic predispositions and environmental factors together influence traits, and sometimes the effect of one depends on the presence of the other. Understanding gene-environment interactions is essential in fields like personalized medicine, disease prevention, and conservation biology, as it helps explain the complexity of phenotypic variation and can guide more effective interventions. You said, phenotypic plasticity, ChatGPT said, ChatGPT. Phenotypic plasticity. Phenotypic plasticity refers to the ability of an organism to alter its phenotype, observable characteristics, in response to changes in its environment. It is a key concept in evolutionary biology that explains how organisms can exhibit different traits under varying environmental conditions, despite having the same genetic makeup. Plasticity enables organisms to adjust to environmental changes such as fluctuations in temperature, availability of food, light, or other ecological factors. This adaptive flexibility can enhance survival and reproduction, particularly in rapidly changing environments. Phenotypic plasticity is not a permanent genetic change but rather an environmentally induced alteration in the expression of traits. Features of phenotypic plasticity: plastic versus genetic variation. Plasticity involves environmentally induced changes in phenotype, which are reversible and do not involve changes in the genetic code. Genetic variation, on the other hand, is inherited and leads to permanent changes in phenotype. Plasticity allows for short-term responses to the environment, while genetic variation offers long-term evolutionary adaptations. Types of sick responses. Developmental plasticity. Changes in phenotype occur during development in response to environmental cues. For example, some plants may develop different leaf shapes depending on the amount of sunlight they receive during early development. Adiplasticity. This involves changes in phenotype that occur during the organism's lifetime in response to environmental conditions but do not involve changes in developmental pathways. For instance, animals may change their metabolic rate or body size in response to seasonal temperature variations. Behavioral plasticity. Organisms may change their behavior in response to environmental changes. For example, animals may adjust their activity patterns or mating behaviors in response to changing environmental factors like food availability or predators. Phenotypic plasticity versus evolutionary change. While phenotypic plasticity can allow for immediate adaptation to environmental challenges, it does not involve genetic evolution. Evolutionary change occurs when the genetic basis of a trait is modified, which may take many generations. Plasticity can sometimes facilitate evolution by allowing organisms to survive in variable environments, increasing the likelihood of genetic adaptation to new conditions over time. Factors influencing phenotypic plasticity. Genetic factors. The extent of phenotypic plasticity often depends on an organism's genetic predisposition. Some genotypes are more plastic than others, meaning that they can exhibit a wider range of phenotypes in response to environmental factors. Environmental factors. Temperature. Many organisms exhibit plastic responses to temperature changes. For example, fish and amphibians can alter their metabolic rate depending on whether the environment is warmer or colder. Resource availability. The availability of food or nutrients can lead to changes in growth patterns or reproductive output in response to changes in the environment. Light. In plants, the amount of sunlight available can influence leaf size, shape, and photosynthetic capacity. Some plants exhibit shade avoidance responses by elongating stems and leaves when they are shaded by neighboring plants. Predators, herbivores. Some species change their behavior or morphology in response to the presence of predators. For example, some plants may develop more toxic chemicals when herbivores are abundant. Trade-offs. Plasticity involves trade-offs, as the ability to adjust to different environmental conditions may come with certain costs. For instance, animals that can alter their body size in response to food availability may sacrifice other traits, such as reproduction or immune function, to allocate energy to growth. Examples of phenotypic plasticity. Plants and environmental cues. A well-known example of phenotypic plasticity in plants is shade avoidance. When plants are grown in low light conditions, they often exhibit taller growth and larger leaves to capture more light. Similarly, some plants change their leaf shape or size based on water availability. Temperature and metabolism in fish. Fish species often exhibit plastic responses to temperature changes. In cold environments, fish may increase their metabolic rate to generate more heat and maintain normal physiological functions, whereas in warmer conditions, their metabolism may decrease to conserve energy. Mating behaviors in animals. In some animal species, mating behavior can be plastic in response to environmental conditions. For example, in some species of birds, males may change the display of their feathers or songs depending on environmental cues such as seasonal variation or the presence of competing males. Coloration in animals. Many species of animals exhibit phenotypic plasticity in terms of color. For example, the rock pocket mouse Chaetodipus intermedius displays different color morphs depending on the substrate in their environment. Mice living on dark volcanic rock tend to have darker fur, whereas those on light sand have lighter fur. This plasticity is advantageous for camouflage and predator avoidance. Cichlid fish and body shape. Cichlid fish exhibit plasticity in body shape in response to environmental factors such as the availability of food and habitat complexity. Cichlids may have more elongated bodies in environments where they are required to move quickly through complex habitats or shorter bodies in areas where food is abundant but more sedentary behavior is necessary. Mechanisms underlying phenotypic plasticity. Environmental sensing and signal transduction. Organisms use environmental sensing mechanisms to detect changes in their surroundings. For instance, plants use phytochrome to sense light changes and adjust their growth accordingly. Similarly, animals may have hormonal or neurological pathways that allow them to respond to environmental stressors. Gene expression changes. Phenotypic plasticity often involves changes in gene expression rather than changes in the underlying genetic code. This can be achieved through mechanisms such as epigenetic modifications, where gene expression is altered without changes to the DNA sequence. These modifications can be reversible and context-dependent. Developmental pathways. In some cases, environmental factors influence developmental pathways that lead to different phenotypic outcomes. For example, the expression of particular genes during embryonic development may be influenced by the environment, leading to changes in morphology or behavior later in life. Hormonal and epigenetic modulation. Hormones are critical in mediating phenotypic plasticity. For instance, changes in the levels of growth hormones can influence growth patterns in response to food availability. Similarly, epigenetic mechanisms can regulate the expression of genes involved in phenotypic plasticity. Costs and benefits of phenotypic plasticity. Benefits. Increased survival. Plasticity allows organisms to respond rapidly to changes in their environment, enhancing their chances of survival in variable conditions. Increased reproductive success. In environments that fluctuate between seasons or in response to changes in food availability, plastic responses can enhance reproductive success by optimizing the organism's phenotype for current conditions. Adaptive flexibility. Plasticity allows organisms to adapt to multiple niches within the environment, providing a buffer against ecological fluctuations. Costs. Energy costs. The ability to exhibit plastic responses can be energetically costly. Allocating energy towards maintaining the potential for plasticity might detract from other functions such as reproduction. Reduced specialization. In some cases, the ability to adjust to various environments might limit an organism's ability to specialize in a particular niche, leading to trade-offs in efficiency or performance in specific conditions. Misleading cues. In some cases, the environmental cues triggering plastic responses may be unreliable, leading to maladaptive phenotypic changes. Practice questions and answers. Question 1, GATE 2022. Phenotypic plasticity in response to environmental changes is best described as a permanent change in an individual. B. A reversible change in an organism's phenotype due to environmental cues. C. The evolution of new traits through genetic mutation. D. An irreversible response to stress. Answer. The correct answer is B. A reversible change in an organism's phenotype due to environmental cues. Phenotypic plasticity refers to reversible changes in an organism's phenotype in response to environmental factors, not permanent genetic alterations. Question 2, GATE 2021. Which of the following is an example of developmental plasticity? A. A bird species changing its migratory behavior in response to food availability. B. A plant growing taller in response to shading from other plants. C. A fish altering its metabolic rate with temperature changes. D. A predator altering its hunting behavior in response to prey density. Answer. The correct answer is B. A plant growing taller in response to shading from other plants. Developmental plasticity involves changes in an organism's development due to environmental cues, such as how a plant may alter its growth form to access more sunlight when shaded by other plants. Summary. Phenotypic plasticity is a crucial adaptive mechanism that allows organisms to survive and reproduce in fluctuating environmental conditions. Adjusting their phenotype in response to environmental cues, organisms can enhance their fitness. While plasticity can offer significant advantages in variable environments, it also comes with costs, such as increased energy expenditure and potential trade-offs. Understanding phenotypic plasticity is essential for fields such as ecology, evolutionary biology, and conservation, where predicting how organisms will respond to environmental changes is crucial for management and conservation efforts. You said, heritability. ChatGPT said, ChatGPT heritability. Heritability is the key constant in genetics and evolutionary biology that quantifies the proportion of the total phenotypic variation in a population that is attributable to genetic variation. It plays a critical